All right. Now, in the cytoplasm, there are all kinds of cyto or cell organelles. So the first one we're going to focus on is the nucleus. The nucleus is very important, and that's because it contains the cell's genetic material, which is DNA. Okay? So when you look at the nucleus, that's kind of what it looks like, right? Spherical structure, and it's enclosed by a double membrane over here, and that's called a nuclear envelope or a nuclear membrane. It's the same thing. So this is the nuclear envelope. And in the nuclear envelope, you probably noticed there are these little pores, openings, right? So the nuclear pores allow things to pass through the nuclear envelope to kind of go in and out of the nucleus. Now, it's very important because there's a lot of exchange in materials between the nucleus and the cytoplasm. For one thing, when we talk about gene expression, you will learn that we actually mentioned this a little bit in the last chapter. You need to transfer the genetic information from DNA to mRNA, right? So it happens inside the nucleus. But the mRNA, the messenger RNA, has to come out of the nucleus and deliver that information to ribosomes in the cytoplasm to produce proteins. So how do messenger RNAs come out of the nucleus? They have to come out through the nuclear pores. Okay, so this structure is actually quite important because with that, things are just going to be stuck in the nucleus. There's no exchange, no information exchange or substance exchange between the nucleus and the cytoplasm. When you look at DNA in the nucleus, they're actually not alone. They actually form a more complex structure with the proteins, which we call the histone proteins. So what you actually see, let's assume you're inside the nucleus, what you can see is actually a complex called chromatin. So this complex consists of both DNA and histones. Remember these are proteins. Now, why do we have a protein component? And that's because DNA molecules are very, very long chains, very, very long. So there's a good chance that they may get tangled, and then they break. So you need to provide some kind of support where the DNA can wrap around and be more organized, and they don't get tangled and they don't get destroyed or damaged. Not destroyed, but damaged. Okay, so that's why we have the histone proteins, and they form this complex called the chromatin. Now, you might not be familiar with the chromatin, but you probably have heard of chromosomes, right? So for humans, we have 23 pairs of chromosomes, and that's a total of 46 chromosomes. So that's what the chromosomes look like. So what's the difference between chromatins and chromosomes? Now, they're really the same thing. They carry the same genetic information. They carry the same DNA. They're just the same thing in different forms, and they usually show up at different stages. Okay, so this is how it works. Usually inside a cell, it's chromatins. Now, when the cell is ready to divide to make new cells, the chromatins will condense and become a compact form. And when that happens, they become chromosomes. All right, so when you look at chromatins, it's kind of very thread-like, right? But when you look at chromosomes, you see all the threads have been really compacted, condensed. So that's chromosomes. So they're really the same thing, but just in different forms. In prokaryotes, the DNA is organized in one circular chromosome. Okay, so if I had to draw, it's just like one circular chromosome. In eukaryotes, chromosomes are more linear structures, right? Linear structure, not circular. And we usually have a specific number for each species. So for humans, then that number is 46 chromosomes. Now for elephants, it will be a different number. For giraffes, again, different number. So the number of chromosomes indicates individual species, different species. Okay, the last structure we're going to talk about for the nucleus is called the nucleolus. So when you look at the nucleus, there is going to be an area that looks a little bit dark. Okay, and this area is where you have aggregations of ribosomal rRNA. Okay, so you see aggregates the ribosomal RNA with associated proteins to assemble the ribosomal subunits. Okay? So this is where the ribosomes are assembled with associated proteins, and these are going to become ribosomes. Yeah. So ribosomes, once they're produced, they come out and they're going to stay in the cytoplasm. And actually, the red dots, those are ribosomes. So these ribosomes are attached to the endoplasmic reticulum. Okay, so these are ribosomes. Now, some of the ribosomes are attached to the ER. Some of the ribosomes may just kind of float freely in the cytoplasm. There's ribosomes, and there are two subunits, right? So you can see this is the large subunit, and this is the small subunit. So ribosome, remember that it's a complex of protein and RNA. And there are two subunits, usually a big one and a small one. Ribosomes synthesize proteins, right? So they will assemble amino acids, put them in a chain-like structure, right? That's the primary structure, the sequence of amino acids, and eventually these polypeptide chains will become proteins. Okay. And this is important information. All cells have ribosomes because all cells need proteins to do different functions, so they have to be able to produce proteins. So all cells will have ribosomes. Now, let's do a quick overview. So different types of cells may have different functions, but for now we learned that all cells will have cell membrane or plasma membrane. Okay? All cells have ribosomes, and all cells have, do you know what's the last one? Right, DNA, the genetic material. Yep. Okay. Next structure, this is important. This is called mitochondria. So this is plural. Singular will be mitochondrion. So D-R-I-O-N. So that's the singular. So the mitochondria are known as the powerhouse or energy factory of a cell. So this is where the cell produces the energy that it's going to utilize. Okay. The fuel that a cell is going to use to burn to get the energy is actually glucose. Very important. So once glucose enters the cell, the mitochondria can break down glucose, break down the chemical bonds in glucose, and that will release energy. Okay, so this is how cell can obtain energy from chemicals from glucose. So that process is called cellular respiration, and we have a chapter that will focus on cellular respiration. So when mitochondria break down glucose and release the energy is usually stored because you might not need the energy immediately, right? So it's just like a battery. You can store the energy in battery and use it later. So the cells are going to store the energy released from glucose, there you go, the energy, to make adenosine triphosphate, which is ATP. So the ATP is like a battery, and this is where the cell stores the energy. But when the cell needs the energy, it will just break down ATP and extract the energy. All right. Okay. Now, what's interesting about mitochondria is that they have their own DNA and ribosomes. All right. Now, inside the cell, the nucleus is not the only place you can find the genetic material. You can find the DNA. Mitochondria are another place that you can find DNA, and they even have their ribosomes. Okay, so that's actually very, very interesting. Okay, next structure, peroxisomes. So peroxisomes are small round organelles, and they are enclosed by a membrane, but it's only a single layer. Okay, so they carry out oxidation reactions. So usually oxidation reactions, you're using oxygen, right? Most times, oxygen to break down some large biomolecules, and in this case, they break down fatty acids and amino acids. All right, so that's their function. Because a lot of times you have these fatty acids, amino acids, they're waste, you don't need them, so you want to break them down, right? Get rid of them and then recycle the different components, like the atoms, and you can use them to make something else. Okay, but the main purpose is to get rid of the waste, and the peroxisome can do the job. So they're kind of like a garbage cleaner, or something similar. Peroxisomes can also detoxify many toxicants. Okay, so for example, when you drink alcohol, now it will be absorbed into your bloodstream, and when it goes through your liver, the liver cells will try to break down alcohol so that it doesn't, it doesn't get or have a toxic impact on other cells. So it's going to be the peroxisomes in liver cells that break down alcohol and detoxify. Okay. A lot of these oxidation reactions will generate a toxic byproduct. Okay, so it is hydrogen peroxide. It's very toxic. So you don't want this to accumulate in the cell to damage the cell. So the enzymes in peroxisomes can safely break down hydrogen peroxide to oxygen and water. So two main functions for a peroxisome. First of all, break down fatty acids and amino acids, right? Get rid of waste. Second, detoxify, right? For example, break down alcohol, break down hydrogen peroxide.